Okay, hello everyone. It is the evening. Let me get my dates right this time. It's the evening of September 28th. How everyone's doing well. So let's just go ahead and dive in for today. First of all, one of my kids is still awake. He had a long nap today. So if you hear background noise from a kid's TV show, I apologize. I hope it's not, invasive for the lecture. I tried to keep it down at a low level. Annabelle Morgana, is the background noise okay? Can you hear me okay? Well, I hope you. Oh, good, excellent. Thank you. Thumbs up. So. Just go over what we'll do today. Quiz 4 and the Urban Ecology Lab are due today on Canvas. And so I think you have until midnight tonight, but they're due today. So I look forward especially to reading the Urban Ecology Lab. I think that I did something very similar as an undergraduate and had a fun time with it. We'll recap on biochemistry briefly, and then we'll dive into a lecture on biological macromolecules. And then I'll spend a good amount of time at the end going over the Today I Am a Microbiologist lab, which is the lab activity for this week, including a tutorial on how to use the portable microscopes. Yes, it is now time, finally, to use the portable microscopes. Yes, we'll save that for the end. And just here at the end, a small change to the syllabus. On October 12th, if you look at the old syllabus, or whichever week that is that corresponds to the week of October 12th, I had a lecture in there scheduled for cellular respiration. I'm not going to do cellular respiration anymore. Especially considering the current epidemic, or pandemic, I should say. I met with a friend of mine on Zoom last Friday. He's a virologist. Thanks for those of you who submitted questions. And we had a really good talk on virology. He gave it, I should say, a very good talk on virology, and he really tied in the COVID-19 pandemic really well. And so that talk that he gave is going to take the place of cellular respiration that was previously scheduled for October 12th. Like I said, it's already been recorded, so I'll probably post it sometime before October 12th, just because we've already recorded it. But I want to hold off a little bit because some of the concepts that he talks about are, at this point, a little bit premature. He talks about things like cell membranes and organelles and ATP, and you may recognize some of those terms from high school biology, but we haven't unpacked them yet in this class. And so I'll probably hold off on posting that lecture until after we've covered some of cell structure and cell function. So maybe this time next week, but still before October 12th. So again, instead of the cellular respiration lecture, which was scheduled for October 12th, we're instead going to have a virology/COVID-19 epidemiology lecture. And that will be posted before October 12th. So on October 12th, I'll probably hang around here on Zoom if anyone wants to have office hours, but I won't be doing a lecture on that particular day. Welcome, Elizabeth. Thanks for coming. So that's, I think cellular respiration is a fascinating topic. And we would normally have a really cool lab tied to that. But because we're not able to meet in person for lab this semester, and because COVID is one of the more important things going on in the world right now globally, it seems very suitable to replace cellular respiration for a virology lecture instead. So I hope you agree with me. I think you will enjoy the lecture that Dr. Beering gave my buddy Scott. Just to recap, on what we just talked about last time. We covered biochemistry, or chemistry in biology. And so just a series of questions. For the folks that are on the call, I will not pressure you to say anything if you don't feel up for it, but if you do, you're absolutely more than welcome. And we'll just kind of carry on. These are just helpful review questions for what we talked about last time. So question 1, a substance with a pH of 8 is what? Acidic, neutral, or basic? Well, when we talked about the pH scale at the very end of lecture last time, and we just broadly said that the pH scale is a bit ironic because it's a scale from 1 to 14. And remember, acids and bases are merely substances that give off hydrogen cations. And so acids release hydrogen cations and bases accept hydrogen cations, positively charged hydrogen. And so with this pH scale of 1 to 14, it's kind of logical to think of things that are higher up on the number scale as being more dangerous, right? Higher is more dangerous, like kind of like temperature, if you will. But it's actually the reverse. Anything that's above 7 is basic, and anything that's below 7 is acidic. So something with a pH of 7 would be basic. And here's just another example of what the pH scale looks like. We'll talk, the reason why we're talking about pH is that this becomes an important concept a little bit later down the line, specifically regarding to how water moves in living systems. pH is an important aspect of that, so that's why we're talking about it. But broadly speaking, anything that has a pH of below neutral, well, this is kind of, below 7 is acidic. So increasing acidity is 0 to 7. And those are things like sulfuric acid, which is of course very dangerous, that would burn your skin, to relatively benign acids like vinegar or lemon juice, things that you can get on your skin and generally be okay. Whereas anything that's above 7 is basic, so relatively benign. Bases are things like baking soda and seawater. But as you get increasing alkalinity, things that are pretty high up on the basic scale, things like lye, which people used to use, for example, to wash their clothes, that can actually hurt you as well. So acidity and alkalinity, it just depends on where you are on the scale. Both can be dangerous and both can be benign. And question 2 of 3. Ions are atoms with what? Well, ions are just atoms, as the question suggests, but they have unequal something. They have a charge associated with them. And so the two subatomic particles that we talked about, again, this is probably not brand new information for anybody, but the subatomic particles that have charge in the context of atoms are protons and electrons. Protons are the positively charged bits, and electrons are the negatively charged bits. And so if you have an unequal distribution of protons versus electrons, whether it's positive or negative, you have an ion. And this is all quite theoretical. Even still to this day, I mean, we talk about atoms kind of offhand. Most people know what atoms are, but they're still very theoretical. Talk to a physicist, they'll probably tell you that's not really how it is. Atoms are kind of these, very, 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 very theoretical things and how they behave. Okay, so in this example, here's a sodium atom that has an equal number of protons and electrons, but if it loses an outer electron, in other words, if it loses a negative subatomic particle, it becomes a sodium cation, which is an ion type, a positively charged sodium. And question 3, which of the following list the types of bonds in order from strongest to weakest. I think this was a question on your quiz. Annabelle, if you don't mind, could you mute?
Oh, sorry.
No worries. Thank you so much. Which of the following list the types of bonds in order from strongest to weakest? And again, this was a question on the quiz this week. And so I think we talked about the 3 bond types, the strongest of which is the type of bonds that share electrons. And so these are covalent bonds. The middling type of strength are the type of bonds where electrons are moved from one atom to another on the basis of electronegativity, which is simply affinity for electrons. Those are ionic bonds. And lastly, we talked about this third type of bonds, which are hydrogen bonds, which are basically attraction between hydrogen, but mostly in the context of water molecules. And that's the weakest type. So C here seems to be the correct answer. Covalent ionic hydrogen in order from strongest to weakest. But just to reemphasize this, just because we're talking about them in terms of their chemical strength does not mean that covalent bonds are the most important from a biological perspective. All of these bonds are extremely important from a biological perspective. Hydrogen bonds may even be the most important, because if hydrogen bonds didn't exist, water would not be able to have the consistency that it does. And as we've talked about, water being the solvent of life, if water didn't have the property of hydrogen bonds, life, complex multicellular life as we know it, probably would not be able to exist on a chemical level without hydrogen bonds. And these other bond types as well. So anyway, there we have it. Today we're going to dive into biological macromolecules, which is found in chapter 3 of your textbook. And so, I know you've seen this same schematic many times throughout the course, but I'll throw it up every chance I get. It's the schematic that we're following in this class. And so, of course, we spent Unit 1 talking about ecology. And then now we're in Unit 2 of the class. We talked last time about biochemistry. So we talked mostly about atoms, and really we built ourselves to how important water is in the context of biology. And today we're going to go a little bit higher, and we're going to talk about macromolecules. So a pretty important thing, and we're going to continue building up as the unit continues to progress. Okay, so just to rehash, chemistry and biology, water is the solvent of life. It's the most numerous, dense aspect of life, if you will. Life is water-based. If you think about life as a solution, the solvent of that solution is water. So it acts as the primary solute for most biological processes. All the other aspects of life, the physical matter of life, the macromolecules, which we'll talk about here shortly, as well as other small things like ions and smaller molecules, they're all floating in water. Okay, so water is the mediator for everything that we're going to talk about going forward. It's extremely important for how life functions. So what are biological macromolecules? Well, let's break it down because that's kind of a mouthful to say. In the Latin, macro means big. Okay, so a macromolecule is just big. Molecule, as we talked about in the last lecture, is simply a group of atoms bonded together. So something like this. This is actually kind of an artist's rendition, of course. Molecules don't really look like these balls and sticks as artists tend to portray them. But they're merely, a molecule is merely a group of atoms that are bonded together. That can be as simple as 2 hydrogen atoms bonded together, or it can be on the order of thousands to millions of atoms bonded together. Molecules can be extremely complicated. So from 2 to many, many, many atoms. Okay, so a macromolecule is simply a very big molecule. And if we keep building up, a biological macromolecule are the macromolecules, the big molecules that are core to living structures. And so if you want to think about life again as a solution, or even to switch tack, think about life as as a Lego set. The macromolecules, the biological macromolecules, are the individual Legos. They're the building blocks for cells, for organs, for organisms, and everything up, going all the way up to ecology and ultimately the biosphere. And so they are really, these biological macromolecules, they're the smallest functional unit of life, of the biotic world. They're the base component. Not, of course, the smallest functional unit of matter, but they're the smallest functional unit of the things that we can begin to think of as life. They are in and of themselves not considered alive. Okay, so if you take out a single Lego from the wider piece, it doesn't really look like much, right? So biological macromolecules, even though they have the word biological in them, by themselves, they're really not considered alive. It's when you start to link them together through various chemical bonds and so forth that we talked about in the last lecture, and they begin to have systems of assembly and function and other things that we'll talk about as we carry on in the course. That's when we start to cross the threshold in things that we call alive, okay? And they have many different functions. I mean, billions of different functions, okay? So there are an incredibly complex group, even though we only are going to posit these 4 broad categories of biological macromolecules. So just to go over it again, biological macromolecules are big molecules that are core to the structure of life. They're the smallest functional unit of life that in and of themselves are not alive, but they are the Lego pieces, the individual Lego pieces, if you will, of life that have many different functions. Okay, so I'm going to go ahead and posit to you that there are 4 types of biological macromolecules. And I'm sure you've heard of most of these, if not all of these, before. We'll explore them in a little bit more detail. So these 4 main types are the carbohydrates, which you can kind of offhand call the sugars, the lipids, which you can offhand call the fats, the proteins, which are a really big group of macromolecules, which are too much for one word. We'll explain why that is when we get there. And then nucleic acids, which is broadly concerned with the genetic material, also a little bit with energy. So of these 4 types of biological macromolecules, each has its own primary subunits, and each has its own specific set of functions. So we're going to explore successively each of these 4 types of biological macromolecules, starting, of course, with the carbohydrates. So carbohydrates, another, an offhand term you can use is the sugars, although that's really a little bit too simple. That's just a kind of common name. Okay, so the carbohydrates, they, again, we break them down into its simplest unit. So the primary subunit of a carbohydrate, if you take a single, we're using the term carbohydrates in the plural here, but if you want to break it down to the singular, a single carbohydrate, that's called a monosaccharide. Mono, of course, maybe the term, the Latin term, simply means one. So a single saccharide, these are the simple sugars. Okay, so a really good example, really the best example, and really the only one we'll talk about throughout the rest of the class, is glucose. And maybe you've heard of glucose. It's a very simple sugar. It's a monosaccharide in and of itself. Here it is, the actual chemical structure of glucose. As you can see, it is a molecule of carbons and hydrogens and oxygens that are actually covalently bonded to one another. So this is where the chemistry and biology comes in. These are covalent bonds. Okay, so this is a single glucose molecule. And maybe you've heard of glucose before. We talk about it a lot in the health field in the context of people who, if you know anyone who has diabetes, for example, we talk a lot about their body's response to glucose and blood sugar. And glucose is an extremely important monosaccharide in the context of living things, because it's the most common monosaccharide that our bodies, multicellular life in general, breaks down for energy use. And so the food you eat ultimately gets broken down into glucose, and then it gets broken down even further into the type of energy that your body uses to fuel its various biological processes. So glucose is extremely important. But that doesn't mean there are other types of monosaccharides. Fructose, for example, which of course you can get if you eat a fruit. Galactose, which is kind of a more highly processed form that you find at other places. We're mostly going to talk about glucose in this class. Again, glucose is a monosaccharide. It's an example of the primary subunit of a carbohydrate. So if you have monosaccharides, as you can imagine, if you can have a singular, you can also have multiple. And so, thankfully, the people who named these different things decided to name it something that makes sense. In this case, polysaccharides. Poly simply means multiple. So if you have multiple monosaccharides all bonded together, and these are covalent bonds or ionic bonds, it just depends. That multiple monosaccharides are all bonded together into a long chain, as is shown right here. That is called a polysaccharide, a chain of many saccharides. And so these, for example, is many glucose molecules that are bonded together. So each individually is a monosaccharide, but the whole thing together is a polysaccharide. And you keep building this chain out. Maybe you've heard of starch before. Starch, it's a polysaccharide that's basically a long chain of glucose molecules. Okay? And starch is an extremely important polysaccharide in the context of life. It forms a lot of structures. It's broken down for energy use, as you know, because of course its base component is glucose. So there are many types of carbohydrates, there are many types of polysaccharides, many types of monosaccharides, but we're mostly going to hone in on glucose. So there is the function, the structure rather, of carbohydrates. It's built with monosaccharides and it forms these long chains called polysaccharides, glucose building a starch polysaccharide being one example. So going into carbohydrate function, which is the other end of the coin. Broadly speaking, this is broad, carbohydrates are involved in short-term energy storage, and this is kind of, you know, what we often talk about. If you're a health-conscious person and you go to the gym and you're conscious about what you eat, there's a lot of talk about managing your carbohydrates, of course. And so if blood sugar, if you are concerned with maintaining your blood sugar, like you're hyperglycemic or hypoglycemic, you're basically talking about glucose content, the concentration of glucose in your bloodstream. Okay, so you really are tying yourself into short-term energy storage. If you're hypoglycemic, for example, I often get them mixed up, but I tend to think of that one as the one where you tend to have low blood sugar. So if you eat a potato or a banana or some pasta, your body can really quickly break that down into glucose and really quickly process it, and you can feel better quite quickly. On the reverse end, if you're hyperglycemic, you have too much glucose in your blood for a variety of reasons, and you maybe don't want to have so many carbohydrates. I digress. So short-term energy storage is one example of how carbohydrates function. Another function is actually structure. If you take many fructose monosaccharides and build them up into a long polysaccharide chain, that polysaccharide chain is called cellulose, which is what most plants are made out of. Cellulose is extremely important for plant structure. It's what gives wood its density, for example. So very, very important. And, we, of course, it's helpful for us if we consume plants. It's a source of fiber that helps our bodies digest things as well. Okay, so short-term energy storage and structure are two very important aspects of how carbohydrates function. And here's a really important thing, and this gets back into how important water is in the context of biology. Carbohydrates, if we consider their chemical attributes, carbohydrates tend to be what's called hydrophilic. So if you break that word down, hydro means water, and philic means affinity. So if you put those together, water affinity or water loving. So carbohydrates, they like water. In other words, they don't shy away from it. In other words, they dissolve easily. So for, if you take a sugar cube, for example, and you drop it into a glass of water and you shake it up, sugar is basically straight glucose or fructose, depending on the type of sugar. And if you put it into water, its base chemical structure is hydrophilic. It loves the water and it dissolves very easily. And so that has some pretty important ramifications in the context of biological processes, which we will talk about as time goes on. But that is enough on carbohydrates for now. So carrying on, the second category of biological macromolecules are called lipids. We'll break the lipids down into two types. The true fats, which I'm only going to acknowledge that they exist. They're important for a variety of reasons. You eat them and they're important for your diet. But then there are the phospholipids, which are very, very important, and we'll spend most of our time talking about them right here. The primary subunits, and again, these are not, I don't expect you to memorize these complex chemical structures by any means. I had to when I took E-course biochemistry. I did not enjoy having to memorize all these structures, and I'm not going to require you to. But this is just more or less for your edification. Lipids have three primary subunits. Glycerol, which is this pink portion up here. Fatty acids, which are these long chains of carbons and hydrogens. And in the context of phospholipids, which are really important, we'll talk mostly about a polar phosphate group, which is shown here in orange in the phospholipids. Okay, so that's the base components of lipids, which I don't expect you to memorize the intricate chemical detail of, but really going into the really more relevant aspect of lipids is how they actually function. Extremely important, extremely important. Okay, so one thing is, in contrast to carbohydrates, which were broadly involved in short-term energy storage, lipids are involved in long-term energy storage. If you're a person like me who gained some weight in the context of the COVID shutdown, I call it the quarantine 50. I'm still working it off. You probably know that we kind of say it. Fats are involved in long-term energy storage. So that's avocados, for example, are really great to have. They keep you full for a long time. And anything that has a lot of fat in it, if you eat a meal that's highly fatty, it will keep you full for a long time. And one of the reasons why is it takes your body a while to break it down, but it will also keep you satiated for a longer period of time because it's a longer term a source of energy relative to carbohydrates. Okay. Another really important function of lipids is the prevention of water loss. And really mostly in the context of the phospholipid bilayer, which is very, very important. I can't emphasize this enough. Probably one of the most important concepts we'll talk about in the entire unit, the phospholipid bilayer. This broadly relates to the tendency of lipids, the chemical tendency of lipids. In contrast, again, to carbohydrates, lipids tend to be hydrophobic. So anytime, if you say you have a phobia of something like arachnophobia, you're afraid of spiders. If you say hydrophobia, that's something that's afraid of water. So something that's afraid of water in a chemical sense does not dissolve. And you broadly know this, right? If you take some olive oil and you pour it into a glass of water, what does not happen? Generally speaking, the water and the oil are not mixed. It has to do with their polar nature. the oil being non-polar and the water being polar, which we'll talk about again a little bit later on. So lipids, in a broad sense, they are hydrophobic. They do not easily break down in the context of water. They do not dissolve, which has extremely important implications in the context of life, because water is the solvent of life. It permeates everything, and that includes aspects of where living systems like lipids are afraid of water. So this phospholipid bilayer that we'll talk about, I think, in the next unit, this is extremely important. I can't emphasize that enough. Please take a hint right here. Extremely important. And if we kind of bump it up to a more higher tier, this is a cross section of a leaf right here. Have you ever felt the outer surface of a leaf, like an elm tree, for example? They tend to feel kind of waxy, right? That's called the cuticle, the outer waxy surface of a leaf. Those are lipids. And because lipids are hydrophobic, they are afraid of water, and so that is a way that plants help prevent water from escaping their leaves, because the water is trapped inside. We'll talk about plant physiology a little bit later on. I have to stop myself whenever plants come up, lest I go off into a tangent. But extremely important, everyone, the hydrophobic nature of lipids. Very, very important. So the third category of biological macromolecules, deep breath here, we're getting close, halfway done, almost there, are the proteins, okay? Proteins, and I'm sure you've heard proteins before, okay? The primary subunit of proteins are called amino acids, okay? Of which there are 20 different kinds. There are 20 kinds of amino acids. When I took biochemistry, which was a class that was required for the biology major as an undergraduate, I had to memorize the structure, the chemical structure of each and every kind of amino acid, all 20 of them, okay? So again, I'm not gonna require you to memorize the chemical structure of every single amino acid, but I do want you to know that amino acids are the primary subunits of proteins and that there are 20 kinds of them. So if you, again, kind of think of amino acids as the base Lego building blocks of proteins, if you have 20 different kinds, I mean, we have 26 letters in the English alphabet, and we can, how many words do we have? You know, tens of thousands of words in the English dictionary. It's a lot. So, you know, if you have 20 different letters, for lack of a better term, letters that build different types of proteins, there are a lot of proteins. And so depending on the particular order of amino acids that are linked together by these bonds called peptide bonds, which are basically protein bonds, you can get these long chains that are called polypeptide chains. And a polypeptide chain is basically a protein. But because there are 20 different types of amino acids, and they can each have, if you think of each one as like a different letter type, and the order of the different words of proteins and sentences, you can get a lot of different types of proteins. So there are a lot of different types of structures involved. Alpha helixes, for example, or beta plated sheets. They get, you know, the structure of proteins are quite intricate. And so if there's an intricate diversity of structures of proteins, as you can imagine, they have a heck of a lot of function. So proteins in your body, they do everything. I mean, in the colloquial, we kind of tend to think of proteins as meat, right? And meat, it's like the muscle, right? That makes up your body. But that's an extreme understatement in terms of what proteins do. Proteins, in terms of their biological function, they do everything, almost everything. There's name some kind of biological process. It doesn't matter how small, doesn't matter how specific. There's probably a protein that does it. New proteins are discovered every year. Okay, so there are many proteins that haven't even been described yet that are new to science. Okay, so proteins, because of how diverse they are in terms of their structure, They do all kinds of things. They do things like maintain structure, like collagen, which is really important for your muscles and your tendons and your hair. They are contractile. They're proteins that are involved, of course, in the movement of your muscles, like myosin and actin. They're involved in things like transport. So hemoglobin, for example, is a protein that you may have heard of before that's extremely important in moving oxygen around in your blood. They are involved in storage, casein, which is a protein that stores protein in milk, really important. Hormones, which are basically chemical messengers that take different hormonal messages from one part of your body to another. Insulin is really important. That it takes messages of how your body maintains glucose levels. So this is kind of tying in carbohydrates and proteins a little bit. None of these categories we're talking about, of course, are mutually exclusive. We are complex multicellular organisms. All of these things we're talking about, proteins, carbohydrates, lipids, they're all interacting with one another in really complex ways. They're not mutually exclusive. Enzymes, which we'll talk about at some length a little bit later on. Protection, helping your body fight foreign substances. Ion pumps, signaling receptors, the list goes on and on. Proteins do it all. They're really important. So they do, a heck of a lot of jobs in our bodies, not merely involved in things like muscles and things like that. The job description of proteins is much more intricate than that. And again, the reason why is because of their intricate chemical structure on the basis of these 20 kinds of amino acids that can form a heck of a lot of different types of proteins. And lastly, the last type of biological macromolecule are called the nucleic acids, of which there are a few different types. We'll talk a lot about them we'll talk a little bit more about them this unit, but mostly next unit, Unit 3. The primary subunits are either ribose or deoxyribose, and I'm intentionally keeping it broad here because we'll talk a lot more about them in Unit 3. Some kind of nitrogenous base, these 5 different kinds. You'll hear a lot about these A's and T's and G's and C's. Don't note them now because they're not important for Unit 2, but they'll become very important for Unit 3. You'll hear this come up again, these nitrogenous bases. And finally, a phosphate group. So here's kind of a schematic of what an individual nucleic acid looks like. A nucleotide, which is, again, a ribose or a deoxyribose. We'll talk about that at some point. Any of 5 different types of nitrogenous base, which is shown here. So an A, a T, a G, or a C, or a U. We'll talk about what that means a little bit later on. And finally, a phosphate group. So all of these things are really important. That's the actual structure of nucleic acids. So you will see this again. Not so relevant in the context of Unit 2. Very important for Unit 3. But really more important for this unit is how nucleic acids function. They are involved in a number of different things. So for one, the proteins that we just got done talking about, the nucleic acids actually code for their synthesis. So what actually tells the body how to take a particular amino acid and link it to another particular amino acid, and what amino acid comes next to build a polypeptide chain that ultimately becomes a protein. The molecules that contain the instructions for how to build these long, complex polypeptide chains, of which there are millions, are actually encoded for in nucleic acids. So these are, RNA and DNA. I'm sure you've heard these before. They're the blueprint for proteins. Extremely important. Okay. So although they may not do the actual cellular work like proteins do, they code for the molecules that do the cellular work. So without them, the proteins wouldn't exist. So pretty important. And also extremely important is that another type of nucleic acid is this thing called ATP, adenosine triphosphate, which is, if you break down glucose over multiple forms through time, it becomes this ATP molecule. Extremely important for how basically all life that we know of, with a couple of weird exceptions, but we'll ignore those for now. All life that we know of, for the most part, uses this specific type of nucleic acid called ATP to fuel the cellular processes. It's the reason why we're able to think and move and be alive, is this ATP. So extremely important nucleic acids are. Okay, so I hope, I know that was kind of quick and a lot. So we're going to continue to rehash these 4 types of biological macromolecules, the carbohydrates, the lipids, the proteins, and the nucleic acids. So that was a quick introduction to them. They will come up again, but I would like you to know broadly the base subunits of each, as well as the base function of each. So now, for the rest of this evening, I really want to hone in on what we would be doing if this were a traditional in-person class, is microscopy. This would be the part of the class where we would be taking out the microscopes from underneath the cabinets, and it's kind of exciting, and you get to look at the microscopes, and it's a lot of people tend to enjoy this aspect of the lab component of the class most, because you get to see some pretty cool stuff under the microscope. Unfortunately, we're not able to do that this semester for obvious reasons, but we're going to come as close as we possibly can to approximate that under the circumstances. And so the discipline of using microscopes to look at really small objects that your naked eye can't see, that's called microscopy. And that's not specific just to the field of biology, of course. Physicists use microscopes, jewelers use microscopes, engineers use microscopes, which is the discipline of microscopy. It's basically using technology to view objects that we cannot see with our eyes, things that are too small for our eyes, okay? So there are a few different types of microscopes. I'm just gonna introduce the 3 most commonly ones, starting actually with the most intricate and most expensive, which you will not use in this course. I've only ever had the opportunity to use one once when I was an undergraduate. So these are electron microscopes. So electron microscopes are still pretty cutting edge. They're not generally available. I don't think you can get them on Amazon. Maybe you can. I haven't looked, but they are pretty sophisticated because they use basically accelerated beams of electrons. They take a very concentrated beam. You have some kind of specimen. You take it, you put it under the focal point, and the electron microscope beams that object really intensely with electrons, and then through a pretty sophisticated computer algorithm, the computer will boop, 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 shoot out on the other end an image, basically an imprint of what it looks like. So when you get an output from an electron microscope, you're not actually viewing the live image of whatever your specimen is. You're looking at an imprint, but because electrons are so small, subatomic particles, and you're beaming a really high intense ray of electrons at your specimen, you're actually able to get amazing resolution, up to 100,000 times magnification. So it's amazing what some electron microscopes are able to do in terms of and how small some of the things that you can see with them. You can see things like atoms, viruses, which are really small, or even the quote-unquote ultrastructure of cells, like things like organelles and even smaller parts of organelles, which we'll talk about a little bit later on, really small parts of cells that you can't see with a typical microscope, okay? So, typically, this is like the base cost of one. They get $40,000 and up. As I said, I only got to use an electron microscope for like 10 minutes. I had to schedule it like a month in advance. When I was an undergraduate, I went to UC. I went to a small college, but I had to schedule it at UC Santa Barbara, and I got to use it to look at some algae specimens when I was doing some undergraduate research. I digress, but it was kind of fun. I got some pretty cool images out of that. But again, you can get some pretty cool stuff out of this. So here's some examples of what you can get with an electron microscope. This is extremely relevant in the context of our current global situation. You may know we commonly call this COVID or COVID-19 Test, test. Can you still hear me? Everyone? Okay, good. Thank you. I was just making sure because I got a weird message. So anyway, SARS-CoV-2 is the causative agent of the coronavirus, the COVID-19 pandemic. And so this is an electron microscope image. It doesn't look like much, but if you think about how small these things are, you know, I don't have much of a scale here, but, you know, maybe something that would fit on the foot of this little creature here. So, you know, some thousandths micrometers small, very, very small. This is actually quite impressive. And the reason why, well, no, I'm actually not going to talk about that because Scott will go into that when we do our virology lecture. And then these things are called the tardigrades, which are like, you know, extremely small little animals, actually. You know, so here's a scale, 100 micrometers, which a micro is, you know, extremely small, is 1/1000th of a meter. So it's like 100th of a millimeter. So it, you know, this is very, very, very small. And that's what they look like. They're kind of freaky looking, but these are really cool organisms. We'll talk a little bit about that near the end of the course. Go ahead and Wikipedia them if you're interested in the meantime. The water bears are a common name. But anyway, this is the kind of stuff you can get with an electron microscope. Unfortunately, you know, BC does not have one. CSUB used to have one, but doesn't anymore. Probably the closest one that we have is over near UC Merced or something. So these are still quite rare. I mean, relatively inaccessible for most folks. More commonly used, and I'm sure maybe you've used one of these before, is a compound light microscope. And so, as the name suggests, light microscope uses a variety of oculars and magnifiers with direct light to directly, basically, take something super small like cells and, you know, shine a bright light on them and you know, use multiple compounded lenses to get a really fine view. Anywhere from 40 times magnification all the way up to 1400 times resolution magnification. And so you're actually, when you're using an electron, or excuse me, a compound light microscope, you're actually viewing the live specimen, unlike an electron microscope. You know, so you're viewing, it's like basically taking a flashlight and zooming in on something very close. And so this is what we would be using if we had our formal lab. So, you know, maybe you've used these before in high school biology. So it uses slides, which are either pre-prepared or you can make your own, called wet mounts. You know, you can view a lot of things this way. You can view bacteria, archaea, which are two of the domains that we'll talk about. You can view multicellular things. And, you know, these are much more available. If you want to get a really good one, you're looking at $1,500, which is still a heck of a lot of money. But still, you know, if you save up, you know, most folks can afford one. You know, I think Amazon will even sell you a cheaper one for under $1,000. But anyway, they're pretty handy compound microscopes. So you can use them to view things, individual cells, like unicellular things like this paramecium here, which is pretty common in lake water, or you can use it to view multicellular organisms like this Elodea here, which is probably the most common type of plant that you put in a fish tank. So these are individual cells that are linked together here. So a multicellular organism or an individual unicellular organism. And finally, what I am able to offer you in the context of this class are portable microscopes, which I asked you to go ahead and buy. These are, you know, these are also light microscopes. Okay, so you know, it's very similar in how they actually work. Actually, very similar to this. Just they don't have nearly as much power. They're much weaker than compound light microscopes. And so, even though you know, Amazon might tell you, or the manufacturer might tell you that you can get up to 1,000 times resolution on one of these, that's kind of it's kind of false advertising, in my opinion. If you had immersion oil or something, which is an oil that can help you focus on this maybe. But generally speaking, in my experience using this over the past few months, I got this back in, I think, well, late last year. I've had most success up to about 100 times magnification. So you can still view things, 100 times is still pretty good. You can do things that are smaller than you can see, but you can't, generally speaking, quite get to the cellular level with these. You can really closely view fine structures of plants and insects and rocks and things like that, but you cannot quite get to this level. They're just not quite powerful enough. But you can still get some pretty cool stuff. So for example, these are things that I've been able to use just in, goofing around in my backyard. This is a bluebird feather. So, this here's the main part right here, but then each individual kind of offshoot here, look at the intricate structure of a feather. It's quite beautiful. And then this is a mite that I saw in my lawn. So the red part here, it's kind of related to spiders. So if you have this surrounding plant is actually a moss. It's in my lawn, but it was a moss in my lawn. So if you have a lawn that's well watered, and if you have any moss growing in your yard, I recommend rooting around in moss. You can find some pretty cool stuff like mites in there. Lawns are a cool place to hang around, but bird feathers as well. Water. If you have ants or an insect, you can anything that you can easily access in the outdoor environment. You can get some pretty cool stuff with this. Okay, so how to actually use this thing, and I'm going to exit screen share here for a second. If you can see me, I've got my portable microscope right here. I know I'm blurry for a second. There we go. I've actually got it right here. If you have yours in hand, feel free to follow along. It comes with a little mount, like a little silver mount that I tend not, I don't think it's that handy. But if you want to use that, you're more than welcome to. Can you still hear me, the folks on the call? Feel free to turn your cameras on and interact if you like, because this can be pretty helpful. Anyway, I'm going to just give me a moment here. What I'm going to do, and I'm actually going to create a separate video that shows you what I see, because I'm limited a little bit by my camera on my computer. But I'm going to take the USB port, and before I even get there, I should say that I gave you the link to this on Amazon, and this particular brand only works with Samsung or Windows. Oh, here's somebody. Welcome, Melba. This particular brand of microscope only works with Windows or Samsung devices. And so if you are an Apple user, if you have an iPhone or a Mac of some kind, unfortunately, this will not work. I am sure there is an equivalent out there, equivalent product that works on a Mac or an iPhone device. I just don't know about them because I don't use Apple devices. If you purchased the $20 one that I recommended for you to use on Amazon, Windows devices and Samsung phones and tablets and things are still extremely common. Ask a friend or a relative or a neighbor to use theirs. That's the one limitation that I can tell you. So my laptop, I'm going to start by showing you what I can actually do with my laptop. And then I'll do a separate video that shows you I'll show you how to do it. You can actually use your phone for this. But let me just unplug this one thing here. Oh, no, I don't need to. Okay, so I'm going to take the USB port on the camera and plug it in to my computer. It works the same way whether you're using your phone or your computer. It works, the actual function of the camera works the same exact way. So I plug it in, and the light turns on. As I said before, this is a light microscope, so you need a source of illumination for for you to be able to get something. And I'm going to go ahead and share screen again here, and share, and oh, come on now, I hope this works. There it is. Okay. Okay, so here we go. So I know this doesn't look like much right now. I am on, if you have a Windows, computer like I do, there is an app that's default in your Windows 10. There is an app that's default on your computer called Camera. All you have to do is come down to the search tab and type in Camera. And this is the app that I'm using. It's default again in Windows. If you're using your phone, there's an app that the camera will actually tell you to download. It's called mScopes. Let me stop share here for a second. If you're using your device, there's an app called mScopes, and if you are a sophisticated technology user, there are other apps you can use, but this is the one that works for me. I'll again do a separate video on how to use your mobile device for this, but let me go back into this because I'm using my computer right now. This is the Camera app. Oh, there we go. Okay, so again, are you seeing what I'm seeing? You should be able to see the surface of my desk here. Good. Okay. So again, you can do. So I'm showing you this through the Camera app, and so hopefully you see my mouse over here, and I can do fun stuff. Like I can take a picture, and I can also, if I come back up here, I can actually do a recording. So if I click record, it should, yeah, start recording for me, and I can actually record what I'm seeing. So again, if you can see me on the screen here, this is the microscope actually doing its thing right here. There's a little gray toggle on here that allows you to zoom in back and forth. Okay, so what I'm going to do is, first of all, I'm going to look at one of my favorite plants that I have mounted on my wall. I'll tell you the story about this in a second. And then I'm going to look at a dead fly that I found on my windowsill. As I said, you can zoom in. You can zoom in on all kinds of fun things. Puddles are great. Your lawn is great. If you find a dead insect, or individual plants are great. They're just fascinating. You can zoom in on all kinds of things. And so, so for example, this is this right here is the leaf surface of what's called the Santa Catalina Island ironwood. So Lionothamnus floribundus subspecies Aspensia, or Lionothamnus floribundus subspecies floribundus, if anyone wants to know. We're actually looking at the surface of the leaf here. This is one of my favorite plants, as the name suggests. This is a plant that's actually in the rose family that is only found on Catalina Island. Has anyone ever been to Catalina Island? This is a tree that you can only find it there in the whole world. And we're looking at the surface of this leaf right here. So these darkish veins that we're seeing, whoop, let me go a little bit further down. These darkish veins that we're seeing are the phloem vessels that the plant used when it was alive to transport sugars. And we can look at the main vein of xylem here that the plant used to transport water across the leaf. See how it's shiny? That's actually reflecting back, but that's actually the waxy cuticle, which are made of lipids. We just talked about lipids a few minutes ago. That's the actual part of the leaf that prevents water from evaporating out from inside. Okay, so we're looking at a close-up of one of my favorite plants right here that's actually mounted on my wall. but I actually have a different plant on this same mount. So this is the Santa Catalina Island ironwood. I have right here, this we're actually looking up closely right now, is the Santa Cruz Island ironwood, which is probably my favorite plant in the world. It's also in the rose family, Lyanothamus floribundus, subspecies Aspensiifolius. So this is actually in the same species as the Catalina Island ironwood, but it's a different subspecies. So it's in the rose family. Look how different it looks. Actually, let me stop sharing here for a second so you can see what these look like in the macro. Well, they're several years old now, so they're a bit broken up, but this is the Santa Catalina Island ironwood, and this is the Santa Cruz Island ironwood. They're the same species, and yet look at how different they are in terms of their leaf morphology. Isn't that fascinating? It's this top plant. When I was out on Santa Cruz Island, I was a pre-med major taking a class in ecology just as a prereq. This was 15 years ago now. I was standing in this grove of Santa Cruz Island ironwoods, and something really deep in me awoke, and I fell in love with botany. And I can't really explain that experience other than to say that trip to Santa Cruz Island converted me away from pre-med and into botany. And so this was the actual leaf that I collected on that trip. It means a heck of a lot to me. So anyway, I'm sorry that I'm getting so passionate here, but let me go back to the camera. So right now we're actually looking at the leaf surface of the Santa Catalina, or excuse me, the Santa Cruz Island ironwood. Doesn't it look so different from the this is the Santa Catalina? Doesn't it look so different? And this is the same species. I just think their morphology, and if we zoom in on it, their anatomy is really different. I mean, their xylem and phloem structure, as you can see, these darker brownish lines, that's quite distinct. Look at these hairs that are running up along the central vein here. That's pretty distinct. That's pretty cool. So I observe that there's these hairs here. I don't really know what they're for. I wonder what they're for. I think that they might have something to do with reflecting the sun to keep the sun away from the leaves so that it can retain water better. I wonder why the xylem structure of this looks so different than this. I think that the xylem structure of this looks to be a little bit more intricate. Maybe it's a bit more efficient than the xylem structure of the one that you find on Santa Cruz Catalina. And so maybe that's a difference in the two different islands having different amounts of water availability. These are just some broad things. I don't really know. I see, I observe, I think. And this kind of reminds me, well, I'm looking at something that's very dear and personal to me. Oh, I also note these interesting little tips here on the ends, which I think maybe has something to do with collecting water. I don't know. Anyway, see these things that I'm noting here? These are you can observe a heck of a lot by looking at this. And so that's actually kind of those are the kind of questions that I would like you to ask. Okay, and let's look at the dead fly. So I have a dead fly here in a jar, and if we, let me see if I can this is going to be a bit harder to get a close-up of, but let's see what I can do. Okay, so it's a bit blurry, but if I take this silver toggle here, and I play with the resolution, I might be able to get it to okay, we're looking at some, there are its eyes. There are its eyes right there. And sorry, it's a bit harder to get a good image here. Bear with me. Okay, so look how hairy flies are. I'm probably not endearing you to flies any more than you already have been in the past. That's actually, see that weird, see this stuff right here? This is actually a spider web, because I think I plucked this from a spider web. And so we're actually seeing the silk right here. That's pretty cool. Oh, look at I don't want to move it. I'm actually going to take a picture. Do you see my mouse here? I don't want to, I'm going to, oh, there we go. at a better position here. I'm actually going to be taking a picture right here of this, and I'm actually going to start. I'm doing a recording already, but I'm on the camera app on my laptop, so I'm actually going to start recording so I can record the actual MP4 file of this specific thing. So I'm actually looking at the eyes. Look at the eye structure there. I wonder why the eye structure is the way it is. I wonder. I observed that the fly is surrounded by spider silk. I'm observing how hairy the fly is. I wonder why. Okay. So anyway, that's enough for that particular recording. So I'm going to stop recording there, and I can actually now pull up the recordings here, and here are the photos. I hope you're seeing what I'm seeing. Oh, okay. This is not live now. This is actually what I was doing a few minutes ago. And I can actually also see the pictures and so forth. Anyway, so getting back. So with that said, are there any questions so far on how to use the portable microscope? I'm going to do a separate video on how to do it. Well, actually, if you'll permit me, I may just do this now, just to spare the complexity. First of all, any questions for the folks on the video? Is that making sense? Is it pretty straightforward on how to actually use it? You can give a thumbs up if you like. Thumbs up. Making sense? I'm just gonna. I may still do a separate video, but I'll quickly show you how to do this on my phone, and which is, I have a Samsung phone. If you have the newest version of Samsung phone, they have a specific USB charging port, which is called USB-C. It's kind of the ovally thing. But this particular camera comes with the old type of charger, as you'll notice. If you break back the USB port here, it's the old type of charger. But they were kind enough to include, if you bought the same version that I bought, it comes with a little adapter here. So if you plug the adapter in like this, now I can plug this into my phone. Okay, so if I plug this in, sometimes I have a difficult time getting it to talk, so I actually prefer to use my Samsung tablet. But let's see. Okay, getting it to talk. Here we go. So now I can actually view images on my phone. And let's see, will it work for me? I may end up doing it on a separate video because right now it's not working. No, it's not working. I'll do this in a separate video because that's easier. It'll be easier that way. But what I just showed you, whether you're using your laptop or your personal device, it works the same way. It's pretty straightforward, the actual how-to of it. So you may have already figured it out. So I'm just showing you. I figured that kind of slowly. It takes me a while to learn these things. So I hope that was beneficial to you. I'm going to go ahead and go back into screen share here really quickly. Talk about lab this week. Okay, so lab this week is that today I am a microbiologist lab, which I think it's called part 1 actually. So what I'd like you to do very broadly is observe 5 different specimens. And so this can be, things that are readily available to you in the outdoor environment. I would like them to be alive. So, nothing that's abiotic. It has to be a living specimen. So it can be a living plant. It can be a dead plant. It can be an insect, whether it's alive or a dead insect. Pet hair. Hair is really interesting. and it doesn't need to be an individual organism either. It can be your lawn. It can be a piece of moss. It can be if you have like a bird bath or something with a bunch of algae growing in it, that can be a really cool place. A puddle, a bird's nest can be really cool. So, basically 5 fields of view, individual organisms or like your lawn. Try to find one of those mites, because I had a lot of fun finding those mites in my yard. They're pretty common. If you have a lawn space, you can find them pretty easily, especially after it's not long after it's been the sprinklers have been on. So anyway, I'd like you to find 5 different specimens, and then use the format of the ABC, which is the written word, the 1, 2, 3, which is an approximation of how big it is, what color it is, and so forth. And then a picture, which is drawing a picture. I'd like you to use the ABC 1, 2, 3 picture format for each of these 5 specimens. Okay, so for each one, I'd like you to note, I notice, I wonder, this reminds me of. So the practice that I was doing just now when I was looking at the ironwoods and the fly is I was just noting things that seemed to stick out to me. I notice things, I wonder about what they do, and this reminds me of. And I'd like you to note as many things as possible. Don't just stick to, okay, I've noted the hairs on the fly, I'm done. Note as many things as you possibly can. The more observations, the better. Effort is really what I'm looking for. The bare minimum is not what I'm looking for. I'm looking for, as much as you can see. You'll notice with these scopes, when you're looking at the micro world, there's a heck of a lot of detail. You can pick up on a lot of different stuff. Small things have a lot of detail to them, so it shouldn't be hard to pick up a lot of different detail, depending on what you're looking at. And so I'd like you to include written words as well as a drawing of each of those 5 specimens in your notebook. And then beyond the I note for each of the 5, beyond the I notice, the I wonder, and the this reminds me of, for each specimen, I'd like you to go a little further in the prescribed format and propose a explanation for what you think is going on, a hypothesis. And so going back to the ironwood example, the different xylem structure that we were noting between the 2 species of ironwoods, I hypothesize that has to do with something with water efficiency between the 2 different habitats, between the 2 different channel islands. Or I hypothesize that the hairs on the fly might have something to do with sensation, so that they can sense where they're going as they fly through the air. And those are just some really broad hypotheses. So I'd like you to include some hypotheses for your observations as well. Okay, so 5 things. You're doing the ABC 1, 2, 3 picture format. I notice, I wonder, this reminds me of. Drawing a picture. What you're noting. Try to explain what's going on, what you think is going on based on what you're observing. And lastly, the last component of this is that just for one, not all 5, but just for one of your specimens, I'd like you to do a recording of anywhere from 2 to 5 minutes. So whether it's an MP4 recording and I showed you how to do that right there on the camera app on the laptop, but you can also do the same thing on your phone. You can use it directly through the camera app on your phone. And so if you record videos on your phone, it works the same way. I'd like you to record a video using the portable microscope where you're kind of narrating what you're seeing. I notice, I wonder, this reminds me of. So I'd like that to be between 2 and 5 minutes of length. You can then upload that MP4 directly to Canvas, or you can post it to some kind of clickable link. An MP4 would be recommended. And then at the end, so okay, I know there's some moving pieces here. You're doing 5 different specimens, and you're doing all this, the ABC, the 1, 2, 3. I notice, I wonder, this reminds me of, a drawing of each, and so forth, and a hypothesis. You're doing a recording of only one, and then at the very end of your actual notebook, I'd like you to do a reflection paragraph on everything, all 5. What you learned, what inspired you, what you took away from it, how the pros and cons of the portable microscope, and so forth. Okay, so I hope that's pretty clear. Are there any questions? Anything unclear about that? Make sense? Yeah. Am I still okay? Good. Okay.
Yes, it makes sense.
Oh, good. Okay. So yeah, and I'm not too concerned with the exact formatting in your actual lab notebook, but I'd still like you to keep to the broad format, which is page number, title, which is today I'm a microbiologist lab, and then your metadata, which is like, okay, the date, the wind speed, the temperature outside, really the outdoor environment, your backyard or a public park or something is the most suitable place to do this. And so the metadata on the top of the page, and then title it something like specimen number 1, a fern in my yard. Specimen number 2, ants. Specimen number 3, my lawn. Or if you have something that you really like about if you have a favorite plant or a favorite vegetable, the surface of vegetables is pretty interesting. Do something that you enjoy, and I would like you to enjoy this. Have fun with it. Try to look at something that you're curious about, and so you'll have your notebook entry, but then also the recording of yourself using your portable microscope. And I'm going to do a separate video, instructional video, showing you, kind of what I see. So that'll be specifically instructing on how to use your mobile device. But that way it'll allow me to show you it from my perspective as opposed to the Zoom thing. So anyway, I hope that makes sense. Thanks for hanging in there for those of you on the recording. Other than that, that's all I have for this evening. I will be live on Zoom again this Wednesday for cell structure. So live at 8:30 p.m., which is Wednesday, the 30th of September. So we're getting pretty close to October. Wow. Okay. Any questions? All make sense? I'll sign out here. Actually, I'm gonna stop the recording. So folks in the recording, see you later.